For joining my lecture today. So we are here talking about the photosynthesis in higher plants. So this is a very important topic in your plant physiology. So this has the maximum weightage in your plant physiology compared to the first two topics that we saw. So we spoke about your mineral nutrition, we spoke about your transport in plants. So compared to all the topics, this has the maximum weightage. So please pay attention to this topic, that is photosynthesis in higher plants. So this, yes, has a lot of equations to remember, but it is not that tough. So you have to remember the net ATP production, so that is very important. So let's see. So the first lecture today we are going to talk about only experiments. So there are some base experiments which you have to remember. So we will remember, or we will talk about the base experiments, and then we will go to the main points. Okay. Yes, Tarini, hi, a very happy good evening. Yes, I'm good, thank you. How are you? So I will get back to your doubts in Telegram after the lecture, okay? Hello, Naveen. Yes, a very happy good evening. So thank you so much for joining my lecture, Naveen. So please do like, share and subscribe. So let's go into today's lecture. So lecture 1, photosynthesis in higher plants. So let's see what are the topics that we have. So what do we know about photosynthesis? So what is our level about photosynthesis? How do you know in photosynthesis plants utilize sun's energy? So how do you know in photosynthesis oxygen is evolved? How do you know carbon dioxide is utilized? Yes, how does it happen? So we have some basic set of experiments. They are called as the early experiments. So today we are going to talk only about the early experiments. Yes, Shimpi, hello, a very happy good evening. Yes, I'm good, thank you. How are you doing? Yes, Dharni, thank you. So we will talk about some of the early experiments, okay? So we have a list of the early experiments where I want you to remember the name of the scientist, the experiment, so what was the experiment they did, and finally what is the conclusion they got. So you have a set of 4-5 experiments which I want you to remember. So today we will talk about them. And where does photosynthesis takes place? So this answer is well known. So the answer is your chloroplast. So we will talk about the structure of chloroplast very soon. How chloroplast is intricately designed to accommodate the process of photosynthesis. So then how many pigments are involved in photosynthesis? So you have really 4 pigments which are involved in photosynthesis, with one being the primary pigment and the rest are your accessory pigments. So we will talk about what is the primary pigment and what are the accessory pigments, what is the function of these accessory pigments. Okay, so what is light reaction? The one that takes place in the presence of light is that light reaction. So how does it happen? So what is this light reaction? So here I might introduce you PS1 and PS2. So this is only one portion of the chapter. Okay, so we are going to discuss all this, but today I guess only we'll be able to discuss the experiments because the experiments are equally important. So you will have questions from the experiments alone also. So at the end of the chapter I have some questions for you as usual. So why should we study about photosynthesis? So yes, plants photosynthesize, plants prepare their own food. So why should we study about photosynthesis? So what good does it do to us? Why should we know? So animals, human beings, they depend on plants for food. Yes, so we are done with our ecosystem, we are done with the biodiversity chapters. So we know that plants are primary producers. Yes. So directly or indirectly, we all depend on plants for food. So though you are a meat eater, so you depend on plants for food. And even if you are a plant eater, normal eater, you depend on plants for the vegetable, the stem, the root, any part of the plant, right? So that is also equally important. Hi Anthony Abhi, how are you doing? Good. So thank you so much for joining my lecture today. So please do like, share and subscribe. So talking about why to study about photosynthesis. So where do plants get their food from? So we know we get our food from plants, and plants are the primary producers. But where does plants get their food from? So green plants synthesize their own food through the process or by the process of photosynthesis, and they are known as your autotrophs. So autotrophs are the group of organisms which can prepare their own food, right? So photosynthesis is a physiochemical process by which it uses light energy to drive the synthesis of organic compounds. So what is the end product of photosynthesis? You and I know. So photosynthesis end product is your glucose. So it is the synthesis of organic molecule. So glucose is a carbon containing molecule, right? So how does plants prepare glucose? By using light energy. So where does this light energy come from? The sun. So this is the process of photosynthesis. So this is how the plants synthesize their own food. Yes, Anthony Abhi, I'm good. Thank you so much. How are you? Okay, so this is photosynthesis. So all living forms are dependent on sunlight for energy, directly or indirectly. So directly I can say I don't need sun because it is so damn hot outside, but directly or indirectly, yes, even I am dependent on sunlight for my energy, because I am in turn dependent on my plants, and plants utilize the energy from the sun to make its own food by the process of photosynthesis, right? So all living organisms, so directly or indirectly, are dependent on sunlight for its energy. So the use of sunlight by plants for photosynthesis forms the basis of light on earth. So imagine if a day goes by without photosynthesis. Okay, so one day is fine. Okay, so if one day there is no sunlight and the plants cannot prepare their own food, so plants can use their stored form of wood. But gradually, if plants stop the process of photosynthesis because sunlight is absent, so ultimately it will affect all of us. So this utilization of sunlight for the process of photosynthesis is the basis of life on earth. Okay, yes, so this releases oxygen into the atmosphere by plants. Now we know that photosynthesis leads to the evolution of oxygen in the atmosphere. But how? How did they find out? Who found this out? How did you know? That we will talk about. So I have a series of experiments. So this chapter talks about the photosynthetic machinery. How does the process of photosynthesis happen? Various reactions that transform light energy into chemical energy. Okay, so this is what this chapter is going to deal about. So remember, this utilization of sun's energy by plants for photosynthesis is basis of all life forms on earth. Fine. Okay, so what do we know? So what are the things that we already know about photosynthesis from your lower class? So photosynthesis is not a new term, right? So you have a lot of, you have heard this many number of times. In your lower classes also you would have read about photosynthesis. Yes, no, Shimpi? Yes, so Chaitali ma'am would have taught about you in your lower class, in your 9th and 10th about photosynthesis. So it is not a new term, it is an old term. So how much do we know about photosynthesis? So does all plants perform photosynthesis or any exception? So all green plants perform photosynthesis. So yes, all plants to some extent perform photosynthesis irrespective of its green or brown. So brown plants like your crotons, yes, they also perform photosynthesis, but the pigment that they have is pretty different. See, you know so much, right? Oxygen we get from water, so not from carbon dioxide during photosynthesis. Yes, you're right. So we have simple experiments which says your chlorophyll, so chlorophyll is the pigment, sun's light and carbon dioxide are required for photosynthesis. Okay, yes, so we know these are the requirements of photosynthesis. So who found this out? So what experiment was done? So once again, the experiments are also important. Experiment 1, to look for the starch formation in 2 leaves. So why are we looking for starch formation in leaf? Yes, so starch, actually glucose is the end product of your photosynthesis, but this is stored in the form of starch in your plants. So if you find out that the plants have starch, it means yes, the plant has performed photosynthesis. So how do we check for the presence of starch? So you take a variegated leaf or leaf and partially cover with black paper and expose it to sunlight. So what is the conclusion? So these are simple experiment. Photosynthesis occurred in only green parts of the leaf. So there you had the where presence of light was there. So you take a leaf, okay, and you cover some portion of the leaf, and then you test for the presence of starch. So if you add iodine, so when iodine turns blue, it means starch is there. Okay, so if you have covered this portion for some time and leave it in the sun, and then take this portion off and add a drop of iodine, the portion that was not covered turns blue quickly, indicating the presence of starch. So why do you test iodine with starch? Because starch has secondary helical structure which can store iodine. So that is the reason, fine. So the surface of the leaf, the portion of the leaf that was exposed to sunlight turned blue in color, indicating that photosynthesis happened in the presence of light. So to look for the starch formation in two leaves. So this was the initial set of experiment, okay. So next, experiment number 2. When a part of leaf is enclosed in a test tube containing some KOH soaked in a cotton plug, the other half is exposed to air. So what is this? If you are not able to understand, so it is how it is. You take one portion of the leaf, okay, so this is the entire green plant. So you take one portion of the leaf and soak it in a glass jar, and this contains KOH. And you tight it with a split cork so that nothing escapes outside. Okay, so now what happens? The setup is placed in light for some time. So only one of the leaf is trapped inside the jar and the other half is exposed to air. Okay, so the setup is placed in light for some time. So what is the conclusion? The leaf exposed to air tested positive for the presence of starch. The other portion that is present inside is not tested positive for starch. So thus this says carbon dioxide is required for photosynthesis. So this is a glass jar, right? So sunlight falls on this as well. So we do not have to prove sunlight is essential, but something apart from sunlight is needed. So what is that? Because this portion tested positive for starch, once again this portion is tested absence for starch. So here, yes, here you're not covering with the black paper, so sunlight falls on the glass jar. So then why can't it make food? Because it needs carbon dioxide. So the carbon dioxide that comes from the atmosphere is absorbed by the KOH. Okay, so that is the reason. So plants require carbon dioxide for photosynthesis and they give out oxygen. So these are two basic experiments which will not make much difference. But now I am going to talk about the series of experiments which is equally important. Yes, you are absolutely right, Shimpi. So KOH absorbs carbon dioxide. Yes, you are right. So we have a set of early experiments. So let's see what are those early experiments. A series of simple experiments which gradually led to the understanding of photosynthesis. So this gave us the basis to understand, okay, so these are the requirements of photosynthesis and this is how it happens. So where does each ingredient come from? So if we are making Maggi, so what is the first thing that we do? We take a vessel, so we take a bowl and you had water. So where does water come from? From the RO tank in your house. Yes, so that is simple. Then what you should do? You should turn on the stove. So you know each and every ingredient has a source from it comes, right? And ultimately where does the taste come from? From the masala that you add. So you know from where which comes from. So similarly these series of experiments made us understand, yes, photosynthesis is a process and how does each component or what are the byproducts of photosynthesis and what enhances the photosynthesis process and what are the essential for photosynthesis. So these are the series of experiments. Okay, yes. So let's see. So experiment 1, so I told you the scientist's name is also important. So experiment 1, the scientist's name is Joseph Priestley. So Joseph, the year is not very important, but the name of the scientist is very, very important. So in 1770, a series of experiments played a very important role, so which talked about the essential role of air in the growth of plants. So if air is not provided, so can plants grow? So that was the question. So how did he find out? So let's see. So Joseph discovered oxygen in 1774. So the same Joseph Priestley, he did a series of experiments. So before that he discovered oxygen. So you do not have to remember the year. So please remember the scientist. So Joseph discovered oxygen. So you will not have direct questions from these experiments, but indirect questions. So which of the following statements are right? Yes ma'am, a very, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay. So candle burning in a closed jar extinguished and mouse suffocated in a closed space. So what happened? So your Joseph Priestley, he took a closed jar and inside the closed jar he placed a burning candle and he placed a mice and he closed it. Okay, so he found that the candle stopped burning or the candle extinguished and the mouse died. So why? What is the reason? So conclusion number 1 was burning candle and the animal breathed the air, but they damaged the air. So both the burning candle as well as the mice, they need air to breathe, but they both damage the air because it is a closed space. Okay, so that is the first conclusion he gave. So now he did a small improvement to the experiment. So the improvement was he placed a mint plant. Any plant, so here he placed a mint plant inside the jar. He found that the mouse was alive and the candle was also continued burning. So conclusion number 2, or he hypothesized that, so the plant restores to air what the burning candle and the breathing animal does or damages to the air. Okay, what the burning candle or the living animal removes from the air. Okay, so this is what he hypothesized. So plants restore to air whatever the breathing animals and the burning candle removes. So this is the hypothesis that he gave. So when he placed a plant inside the closed space, he found out that the candle continued burning and the mice also lived. Yes sir, good evening, a very, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So this is the experiment 1. So please remember the name of scientist is Joseph Priestley. So Joseph also discovered oxygen. So what was the experiment he did? He found out that the burning candle and the animal damaged the air. So when he placed a plant, they were fine. They were perfectly fine. So what was his hypothesis? So in this case, I wanted to remember the name of the scientist, the conclusion 1 as well as the hypothesis. So plants restore to air whatever breathing animals sorry, and the burning candle removes. Fine. So second set of experiments, so this is 2A, so you have 2B as well. So the scientist who did the experiment is Jan Eigenhaus. Okay, so he is a second scientist. So he did a similar experiment as done by Priestley. But he placed the jar once in dark and once in sunlight. So conclusion, he found out that sunlight is essential to plant process. They purify the air fouled by the burning candle and the breathing animal. So that is what he did. So what he did? So he created a similar setup as done by Priestley, but he placed once in dark and once in sunlight. He found that sunlight is needed to purify the plant. Yes, the plant restores to air whatever these animals and the burning candle damages, but someone also has to help the plant grow, right? So that was done by the sunlight. So the conclusion, so sunlight is essential to plant process. So purify air fouled by the burning candle and the breathing animal. So remember, scientist number 2 is Jan Eigenhaus. So he did a similar experiment to your Priestley, and what was his conclusion? So sunlight is essential to plant process. Okay, so experiment 2b. So an elegant experiment where aquatic plants were made to use bright sunlight. So small bubbles formed around the green parts which were not seen in dark. So during the daytime, the aquatic plants had green bubbles formed around the green parts of the plant. So these small bubbles were oxygen. Okay, so the conclusion is the green parts of plants release oxygen. So pay attention. So the second experiment was performed by the same scientist, but he gave us two different conclusions. So what are they? The first one, so the name of the scientist is Jan Eigenhaus, and the first conclusion that he gave was sunlight is essential to plant process without any doubt. The second conclusion that he gave was the green parts of the plants releases oxygen. So this was the second conclusion that he gave. Okay? Yes? No doubt still here? Yes, so what he did, he placed the entire setup in light and he placed the entire setup in dark, and then he found out if there is any change. So he found out that sunlight is essential to keep the plants safe, and the plants continue purifying or restoring whatever the burning candle and the animal damages. Okay, so this is what he found out. Fine? Yes, no doubt. So this is experiment number 2. So what was the first experiment? So let's make a list. So if you are making notes, so write down like this. So first experiment, the name of the scientist is Joseph Priestley. So what he found out? He found out oxygen. And what was the next conclusion he gave? So the conclusion 1 he found out. So your burning candle and the breathing animal, they damage the air. And what did he hypothesize? He hypothesized that plants restore whatever the damage is done by the burning candle and the living mice, right? So he found out that plants restore air. Okay, so this is what he found out. So the second scientist, his name is Jan Eigenhaus. So what he did, so he took out 2 experiments, right? So the first experiment that said was sunlight is essential for photosynthesis. So sunlight is needed for the process of photosynthesis. So you should know the name of the experiment, at least the conclusion of the experiment and the name of the scientist, which is equally important. So the next one, he found out that oxygen is released during photosynthesis. Oxygen is released by plants. So this is what he found out. So 2 experiments are done. Yes, so we have 3 more. So experiment number 3, the scientist is Julius von Sachs. So he was the scientist. Okay, so what he did, he provided evidence for the production of glucose when the plants grow. A small plant will not be able to photosynthesize, but until the plant is not able to photosynthesize, its food is provided by the growing seedlings. So fine. So when he found out that evidence for the production of glucose when the plants start growing, so that is what Julius von Sachs did. So he also told us that the glucose is stored as starch, and starch is the storage form in your plants. So later the experiments, he proved that green substance called chlorophyll, so now we know it as chlorophyll, but he called them as green substance, are present in plants, and they are stored in special bodies within a plant cell, that is chloroplast. So today we know the green substance are chlorophyll and they are stored inside your chloroplast. Okay, so conclusion, so he found out that in green plants glucose is made and they are stored as starch. So these series of experiments gave us the conclusion or the theory of photosynthesis that we know today. So experiment number 3, so the name of the scientist is Julius von Sachs. What did he find out? So he found out that glucose is made by plants and they are mostly stored as starch. So this is the conclusion that he gave us. So once again, let's go back and write down. So we spoke about two experiments. So this is A and this is B. So experiment number 3. So what was the experiment number 3? So it was done by Julius von Sachs. So what did he find out? So glucose is made by plants and they are usually stored as starch. So this is the conclusion that your Julius von Sachs gave us. Okay, so moving forward to experiment number 4. So scientist is T.W. Engelmann. So he was the next scientist. The first scientist is Priestley. The second scientist was your Jan Eigenhaus. The third scientist was Julius von Sachs. And the fourth scientist was T.W. Engelmann. Using the prism, he split light into the spectral components and eliminated the green algae, that is your Chlorophora, and placed a suspension of aerobic bacteria. So what he did was, he used a spectrum, so he split the components of light, and when he illuminated the light, he placed a green algae. So green algae contains chlorophyll, so whichever wavelength it can emit or it can reflect or absorb light, the chlorophyll pigment, so there your green algae will start synthesizing food. So by the process of photosynthesis, oxygen will be evolved. So he placed a suspension of aerobic bacteria. So wherever your algae is getting illuminated and producing food, so there you have the release of oxygen. So there is where your bacteria will get accumulated. So that was the experiment performed by T.W. Engelmann. So the bacteria is used to detect the sites of oxygen evolution. So bacteria accumulates mainly in the blue and the red region of light of the split spectrum, because that is where you have the observable pigment of your chlorophyll. The first action spectra of photosynthesis was thus described. So this resembles roughly the absorption spectra of chlorophyll A and B. So this is where both of these are observed. Yes, okay. Yes, Safdar. Mohammed Safdar, yes, hello. So thank you so much for joining my session. So please do like, share and subscribe. So this is what your experiment 4 says. So this is, yes, Eid Mubarak. So same to you. So thank you for wishing all of us. So experiment number 4, the name of the scientist is T.W. Engelmann. Okay. So this is 4th experiment. So this was performed by T.W. Engelmann. So what he did, so he used a spectra of light colors and he illuminated and placed a green algae. The name of the green algae or the group of green algae is Cladophora, and he used a group of a suspension of aerobic bacteria to detect the signs of oxygen evolution. Yes, done. So experiment number 5, so the last experiment I guess. So by the middle of 19th century, so the key features of photosynthesis were understood. So we understood what are the needs of photosynthesis and what photosynthesis gives us. So all these were understood, okay? Yes. So this is by the mid of the 19th century. So plants could use light energy to make carbon dioxide from water and plants could use light energy to make carbohydrates from your carbon dioxide and water. So the empirical equation, the total process of photosynthesis for oxygen evolving organism is this. So for the process of photosynthesis, you need your carbon dioxide plus you need your water. In the presence of light, it produces glucose as well as it releases oxygen. So this is glucose. CH2ON plus O2. So this is C6H12O6. So this is your glucose molecule. Fine. So you need carbon dioxide. Then you have water. Yes. So for photosynthesis also the plant utilizes water. So this carbon dioxide comes from the atmosphere. So water from the soil. So sunlight. So this gives to the release of glucose plus oxygen. So CH2ON is represented as a carbohydrate. So it is a glucose which is the 6th carbon molecule. Fine? Yes. So experiment 5, so not 6, so this is experiment 5. So the milestone contribution which gave us the understanding of photosynthesis. So this is the milestone experiment, the last of last experiment. So scientist named Cornelius Van Niel. So he was our 5th scientist. So once again the name of the first scientist was your John Priestley, the second scientist was your Jan Eigenhaus, the third scientist was Julius von Sachs, the fourth scientist was T.W. Engelmann, and now talking about the 5th scientist, this is Cornelius Van Niel. Okay, so he studied purple green bacteria. He demonstrated that photosynthesis is a light dependent reaction. So what good does this give us? So hydrogen, which is suitably oxidizable compound, it reduces carbon dioxide to carbohydrates. So though I said carbon dioxide is getting reduced to carbohydrate, there should be someone to oxidize this carbon dioxide. Here the one that oxidizes carbon dioxide is your water. So that is why you have oxygen being released. You have a lot of chemosynthetic bacteria which can also perform photosynthesis. Their oxidizable substance is H2S. So that is why they release sulfur instead of oxygen. So there should be a compound which can oxidize your carbon dioxide. So here in the normal case, the one that oxidizes your carbon dioxide is water. So hydrogen is the suitable oxidizable compound that reduces carbon dioxide to carbohydrate. Yes, this is quota coaching. Fine. So green plants, water is the hydrogen donor and it is oxidized to hydrogen. Some organisms do not release oxygen during photosynthesis. Why? Because their hydrogen donor is your H2S in case of your green sulfur bacteria. So instead of oxygen, you have your sulfate being produced. So the conclusion that Cornelius Van Niel gave us is that oxygen evolved by green plant comes from water and not from carbon dioxide. So this was later proved by a radioisotope technique and your radioactive carbon was used. So this is your C14 carbon. So 6 carbon dioxide plus 12 molecules of water gives you 1 molecule of glucose plus 6 molecules of water plus 6 molecules of oxygen. So this oxygen comes from the water molecule and not from the carbon dioxide. So why do you want hydrogen donor? Because there should be someone who can oxidize your carbon dioxide so that it can be converted into glucose. So this is your hydrogen donor. So who's the hydrogen donor here? Water provides the hydrogen. Only hydrogen can oxidize carbon dioxide to glucose. So here your hydrogen donor is water. In case of green sulfur bacteria, the hydrogen donor is H2S. Okay. Yes, Nushrag, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Yes, I told you this is Kota coaching. Sorry, this is coaching from Kota. So we are doing live telecast from Kota. Okay. Yes, Dharani. So this is experiment number 5. So Cornelius Van Niel, he said that the oxygen evolved by green plant comes from water and not from carbon dioxide. Okay, is this clear? So shall we go back and write down whatever we studied so far? So here I have the 4th experiment. So now we are writing the 5th experiment. So this is by Van Niel. So this is the breaking or the substantially important experiment. He said, so oxygen evolved comes from water and not from carbon dioxide. Done? Is this clear? So all the 5 experiments are clear. So please go through. So make a quick note of all the 5 experiments. So that is equally important. Come on. Should I revise once again? Shall we write down the first 3 experiments? Okay. So experiment 1. So Joseph Priestley performed experiment number 1. So what he did, he discovered oxygen, and what else he told you? So your animals, we damage the air. Okay, so air is damaged by the burning candle and the breathing animal, but what did he hypothesize? So plants restore whatever we damage the air. So this was his conclusion. So experiment number 2 was done by Jan Eigenhaus. So what did Jan Eigenhaus tell us? He gave us 2 conclusions. The first conclusion, he said sunlight is essential for photosynthesis, and number 2, so green plants release or evolve oxygen during photosynthesis. Okay, so you were disconnected. Okay, written all things. Okay, fine, very good. So this is the second experiment. So third experiment was your Julius von Sachs. So what did he say? So he said that plants produce glucose, and these glucose are stored in the form of starch in your green plants. He found out that green substances called chlorophyll is present within special bodies inside your plant cell. They are called as chloroplast. So these are the set of early experiments which are very important. So please make a note of them. You do not have to remember the year, but you have to remember the order in which it was performed. So this will give you the timeline in which it was done. Okay, yes. So shall we go forward to the next part? So moving forward, so the early experiments, so this is all we are done. Okay, so where does photosynthesis occur? So this I guess all of you know. So photosynthesis occurs in the leaf, so especially in the chloroplast. So in anatomy, is year will come, ma'am. So what is it? No, years will not come. So they will not ask you years. They will only ask you the name of the scientist. Okay, Antonia B? Yes? Okay. So we are yet to talk about the structure of your dicot leaf and your monocot leaf in anatomy. So which I will cover tomorrow. So as of now, remember, so leaves contain chloroplast because leaves are the sites of photosynthesis. So it also takes place in other green parts of the plant, sometimes stem. So example is the mesophyll cells in all leaves. They contain a number of chloroplasts and they get aligned along the wall to get the optimum independent light. Okay? Yes? Yes, you're right. In chloroplast, chlorophyll is present. You're absolutely right. So if this is a mesophyll cell, so your chloroplasts get aligned at the space where they can get optimum light, because only light can make the pigment activated, right? So that is the reason they get aligned properly. So can you see? So this is your mesophyll cell. So all these are your chlorophyll. So the green color ones are your chlorophyll. They get aligned along the walls. So they get optimum quantity of independent light so that they can do maximum photosynthesis process. So how is the structure of chlorophyll? Sorry, how is the structure of chloroplast? Yes, Sabu, so you were listening, so you were a quiet audience today. So thank you so much for joining my lecture. So please do like, share and subscribe. So now let's talk about the structure of the chloroplast. So how is the chloroplast designed? So we all know inside the chloroplast we have the chlorophyll pigment. So what is the structure of chloroplast? So chloroplast is a double-walled organelle. So this is the outer membrane. So this is the inner membrane. So this is the intermembrane space. So the space between the two membranes. Okay. So the cytoplasm of the chloroplast is known as your stoma. So in case of mitochondria, the cytoplasm is called as matrix. So here it is called as stroma. Okay. So you have flattened disc-shaped structures. So each of the structure is called as a thylakoid. So when thylakoids are stacked over one another, so this is known as your granum. Okay, so the plural, if you have many stacks like this, so this is known as, so you have more than one stack, so it is known as grana. Fine. So there is a connection between the granum. So this connection is known as your lamellae. Fine. So the space inside your thylakoid is known as your lumen. So the structure of the chloroplast is very important, because if you talk about each of the process, then the location is equally important. Fine. So once again, so chloroplast contains your chlorophyll. So it is a double membraned organelle. So you have an outer membrane, you have an inner membrane. So in between the outer membrane and the inner membrane, you have the intermembrane space. Okay, so the cytoplasm of the chloroplast is called as your stroma. So you have flat disc-shaped structure that is called as thylakoid. So the space inside the thylakoid is called as lumen. So stacks of thylakoid is known as granum, which is singular, and grana is plural. So two granum are connected via your stromal lamellae. So this space is your stroma, right? So you have the lamellae. So this is known as stromal lamellae. Okay, is this clear? Yes, no doubt. So your dark reactions takes place inside the stoma and light reaction takes place inside the thylakoid. So please make a note of it. So you have light reactions and dark reaction. So light reaction takes place inside the thylakoids. So dark reaction takes place in the stroma. So what are light reactions? So when they directly utilize light energy and you have products formed, so that is light reactions. So these utilize the products of light reactions, so they are given the contrasting name, that is dark reactions. Okay, so once again the structure of chloroplast is very, very important. Fine. What about the fee structure? Okay, so you have a support at CPIL. So you have the customer support system. So you can talk to them as to which course you want to take. So this class is free. So all the class that comes in YouTube are free. You don't have to pay anything. So you can talk to the support team at CPIL. So they will call you. So whichever course you want to purchase, so you can purchase. So the fees varies accordingly. Okay, yes. So this is the structure. So this is what I was talking about. So you have an outer membrane, you have an intermembrane space, you have an inner membrane, then you have stroma. For biology, so I don't know if they have anything specific for biology, but it is a package PCB. So yes, I guess biology also is there, microcourses are there, so you can talk to the customer care team. Okay, so the mail ID for you, I am writing it here. So it is, everything is small, so support@cpil.in. Okay, yes. Fine. So the outer membrane, inner membrane, you have the space between both. The cytoplasm is known as stroma. So you have flattened disc-shaped structures called thylakoid. The space inside your thylakoid is known as lumen. So stacks of thylakoid is known as granum, and then you have two granum joined together by lamellae. I know you don't have the number, so that is why I gave you the mail ID. So please mail to this, so they will call you back. Okay? Yes, so this is the structure of your chloroplast. So you have clear division of labor in your chloroplast. So the membrane system is responsible for trapping light energy. You have the outer membrane and the inner membrane. So the membrane system is responsible for trapping the light energy and the synthesis of ATP and NADPH. Okay, so the stroma is where the enzymatic reactions are taking place. So they synthesize sugars which are later converted to your starch. So synthesis of sugar happens in your stroma. Fine. So the former directly is light driven. So the membrane system is directly light driven, that is your thylakoid. So that is where your light energy takes place. Okay, and the later where your sugar is produced, so that is known as dark reactions, because they are dependent on the products of your light reaction. Fine, is this clear? So can we solve some questions now till whatever I have taught you? Yes, thank you. Yes, you're welcome. So now let's solve some questions. So I have a set of 10 questions. Let's see if you understand, if you have understood what I have taught so long. So question number 1. So oxygen which is liberated during photosynthesis comes from carbon dioxide, water, chlorophyll, phosphoglyceric acid. Yes, so what is your answer here? Quick. Yes, what is your answer? Oxygen which is liberated during photosynthesis comes from carbon dioxide, water, chlorophyll or phosphoglyceric acid. Yes, good. It comes from water, so all of you are right. Great. So question number 2, the process of taking in carbon dioxide by plants and releasing oxygen is termed as transpiration, respiration, photosynthesis or endosmosis. Yes, what is the answer? Yes, Antonia B, you're right. For the first question, the answer is B. So the process of taking in carbon dioxide by plants and releasing oxygen is termed as, yes, perfectly right, it is termed as photosynthesis. So transpiration is the loss of water. So respiration is where your oxygen is being utilized and your ATP is being produced, but your photosynthesis, oxygen is being produced. So endosmosis, endo is moving inside. So when water moves inside the cell by the process of osmosis, that is called as endosmosis. So here your answer is photosynthesis. So question number 3, in which of the following process the light energy is converted into chemical energy? So digestive action, respiration, photosynthesis or fermentation. Yes, what is your answer? Yes, what is your answer? Yes, once again the answer is photosynthesis. Yes, you guys are right. So why, Mohammed? Answer is respiration. So light energy is converted. So sunlight is converted to chemical energy, right? So this is photosynthesis. So it is not respiration. So question number 4. So during photosynthesis, oxygen in glucose comes from. So pay attention. You have carbon dioxide plus water, so which gives you C6H12O6 plus oxygen. So this oxygen comes from water. So this is understood. So this oxygen comes from where? So this is the question. Water, carbon dioxide, oxygen, both from water and carbon dioxide. No, that is photosynthesis. So respiration, you do not use light energy, right? So respiration takes in the energy that comes from photosynthesis. Yes, what is the answer? No. So this oxygen comes from water. This is understood. They are asking the oxygen in glucose. So this oxygen comes from where? So both from water and carbon dioxide. So that is your answer? No, directly from carbon dioxide. So hydrogen oxidizes carbon dioxide to produce your glucose. So it directly comes from your carbon dioxide. So your answer here is carbon dioxide. Okay? Yes? It is not both. The answer is B. That is only carbon dioxide. Okay? So question number 5. So photosynthesis is most active in sunlight, yellow light, red light, green light. Yes? Okay, fine. But this is how the questions can be pretty tricky. So photosynthesis is most active in sunlight, yellow light, red light or green light. Yes, what is the answer? Yes, so it is most active in sunlight. So this is the easiest of questions. So question number 6. So if the carbon dioxide content of the atmosphere is as high as 300 parts per million, all the plants would be killed. The plants would not grow properly. Plants would grow for some time and then die. So plants could thrive well. So this portion I have not taught you, okay? But what do you think is the answer? If you have maximum carbon dioxide in the atmosphere, so carbon dioxide is one of the limiting factors of photosynthesis. We will see that towards the end of the chapter. But if you have as high as 300 parts per million of carbon dioxide, so what will happen? Yes. Shimpi says plants would grow for some time and then die. So why would they die, Shimpi? Why would they die? So what will happen if you have more amount of carbon dioxide? So we have more amount of carbon dioxide and less amount of oxygen, right? Yes, what is the answer? Yes, so plants would thrive well, so more the amount of carbon dioxide, so more it can produce your C6H12O6 in the presence of water and in the presence of sunlight, right? So your answer is D, so plants would thrive well, okay? Yes. So question number 7, the isotope of a carbon used extensively for studies in photosynthesis is? It's okay, Shimpi, don't be sorry. So what is the isotope of carbon? So I give you the answer for this question. So if it's C13, so it is like this, C13, C14, C15 or C16. So which one? So which one? C16. So this is for the previous question or this question. So I told you, so later your Van Niel's experiment was proved by your radioisotopes as well, and they use C14 carbon. So option B is your answer. Okay, Shimpi? Yes. So moving forward to question number 8, the discovery of Emerson's effect has already shown the existence of. So Emerson's effect showed that your chlorophyll can illuminate in the red light region. The chlorophyll illuminates in the red light region, and his experiment is also known as red drop experiment. So his experiments have already shown the existence of two photosystems. So we have PS1 and PS2, which we'll talk about in the next class. So PS1 and PS2, so please make a note of it. Emerson's effect is also known as red drop effect. Okay. No, it is option A. So we have two distinct photosystems. So that is what it was discovered. So question number 9. So name the scientist who first discovered that plants purify foul air by your bell jar experiment. Will Slater, Robert Hooke, Priestley, Lean Seniberg. So who first proved that plants purify the foul air by the bell jar experiment? Yes, give me. So this is an easy one. So this is an easy one. Your lectures are very beautiful and helpful. So thank you so much, Sabu. So if you do not understand, so please do stop me right there. Okay. Yes. So it is John Priestley. So perfectly right. So question number 10. Of the total amount of water absorbed by plants, its actual percentage used during photosynthesis is how much? Yes, what is the answer? So 1%, yes, of the total amount of water absorbed by plants, the minimum amount is being utilized for photosynthesis. Yes, perfectly right. So this was the last question for the evening, and I gave you the first set of topics for today's class. So in the next class, we will talk about PS1, PS2, what is cyclic phosphorylation, what is non-cyclic photophosphorylation, and what is chemi-osmotic hypothesis. Okay, so if you genuinely liked my lecture, so please do like, share and subscribe. So today we are not meeting in the evening at 6:30, but tomorrow we are meeting at 4 p.m. We are going to finish your organism and population. So I will bring a set of questions as well. So please do join me for tomorrow's lecture at 4 p.m. So until then, this is me signing off. So please stay safe, take care, and have a great evening ahead. So bye-bye. Yes, thank you, Naveen. Bye-bye.